Well, Dr. Antony, thank you very much for joining me at the ISNTD D3 2016 conference. And you're here from the Royal Tropical Institute of the Netherlands.
Yes, that's correct.
Okay, I was just wondering if you could just give us a quick summary of the work that you're doing at the Institute there and how that's impacting the diseasescape of TB.
Right, okay, so we have a small TB group of 6 people who've been working on the development and implementation of diagnostic assays. Diagnostic assays not only to detect infection with TB, but of course, as you know, TB treatment is extremely complicated and long. So to support the treatment, so to monitor the treatment, detect drug resistance, detect failing cases, and hopefully give clinical information on how to manage those failing cases and what might be going wrong and what action needs to be taken to result in a cure.
Great. And you mentioned in your talk previously about the limited access that the majority of the drug-resistant cases have to diagnostics and treatment. What's specific about your assays that you're developing that perhaps makes them any more accessible, if there is?
Okay, so classically the detection of drug resistance in tuberculosis has been based on growing the organism and testing its susceptibility in vitro in a culture media. And TB is a dangerous organism which grows very slowly, so that puts a huge burden on the laboratory and on the laboratory infrastructure, to be honest, even in Western countries. So if you have limited capacity, then you have big problems. There have been significant advances, of course, with molecular biology, and you all know the Cepheid Gene Expert cartridge can do a primary screen for rifampicin resistance, which is a good marker for multi-drug resistant phenotype of the organism. But in and of itself, knowing that there is a rifampicin resistance in the organism is not enough to inform the second-line treatment, which is necessary. Mm-hmm. So you still need, at the moment, to culture the organism, although the new drug regimens and new developments will hopefully result in some effective, standardized, more tolerable, because the second-line drugs are more toxic, regimens. What we are looking at is to try and do, conceptually, you could look at it a way of checking the activity of the drug in the patient. So we want to look at a clinical response in the patient, and we want to measure objectively, easily measurable markers, circulating markers. So we look at circulating immunomodulatory markers in the serum of the patients, and we look at their kinetics over the course of the treatment. And if there is a response which follows our known curves, then we say that the patient is responding to the therapy, everything's going well, it's all under control. If something is unusual or the markers are going in the wrong direction, then you can say the patient is not responding to the therapy. Of course, these markers will not in and of themselves tell you why that response is not occurring. So it could be because the patient has forgotten to take the drugs. The drugs which they were supplied with were for some reason inactive. Maybe they were switched for fake medicines, or they've been stored for too long at the wrong temperature in the pharmacy, or, which is what you screen for with the in vitro assays, or the bacterium that they're infected with is no longer susceptible to the drugs because it's generated resistance. Now, if you can identify this subset of patients who are not responding, of course you can do a clinical and laboratory investigation focused on those patients and leverage what infrastructure you do have towards those patients. So that's a bit our concept.
Great, thank you. So moving forward with that assay, do you think there's any potential scope for an RDT to come from that?
Absolutely, because that's quite an important point. We've selected a biomarker which is quite highly expressed, which has, I mentioned in my talk, it's been repeatedly one of the most informative biomarkers from a series of high throughput and high and targeted biomarker studies, which gives us confidence, because if a biomarker comes out in a single study, should be a little bit cautious, even if it's your own study. It's expressed at a high level. It's measurable by commercially available ELISA assays, and we heard in other talks in this meeting today that there is quite a lot of exciting activity on automated quantitative ELISA assays. There's upconverting phosphors, there's Philips have developed an assay. We heard from Professor Rosanna Peeling that there's a mobile phone attached device. We saw a slide of that, which can apparently perform an ELISA assay on a mobile phone. So indeed, this is one of the reasons why we're focusing on this, because we really believe that there can be a point-of-care device to measure this. Yeah.
Great. That brings me on to my next question. That you mentioned that it's widely used as a biomarker, and you also mentioned that right at the end of your talk that your assay would be useful in high-risk groups, so to asymptomatic children who are...
Well, not asymptomatic children, because children have more propensity to develop disseminated tuberculosis. And as you know, most tuberculosis is diagnosed from sputum, and children often don't have lung TB. They don't produce sputum with mycobacteria in, and diagnostics in children is a huge problem. Also very dangerous, because TB meningitis is often a medical emergency. Well, it is a medical emergency, but it's more common in children. So because we're looking at a systemic effect and the kinetics of response to treatment, we believe that this approach could be applicable and should be investigated in those more difficult cases. And that is exactly what you were suggesting. The extrapulmonary adult cases, of course, are also a diagnostic challenge, because most TB diagnostics is currently based on detection of mycobacteria in sputum.
Yeah. I also just wanted to ask if you had any ideas how you would be able to overcome a potential issue regarding distinction between other respiratory diseases in children. Yeah. And active TB. Absolutely,
and that's the killer question. That also goes for the assay. So we also, I mean, of course, we also heard today that people are most content when they can see the organism, so they're most content with microscopy, a little bit reasonably happy, but a little bit less happy with culture. You can contaminate cultures, you can contaminate PCRs, so DNA, and then as you move towards microbial derived antigens or host derived markers, there is less and less confidence. So our proposal is indeed that these markers would not be suitable alone as diagnostics, but when you link them to the kinetics of treatment, and the literature is accumulating more and more evidence that this is the case, then you will add an extra level of specificity. So it might never be 100%. Of course, you could have somebody who coincidentally, but nothing is 100%. So we believe you add the specificity by linking the intervention with the kinetics, and the intervention is starting treatment.
Great. And last question, I promise. Your work is really exciting in terms of how much it can contribute to the monitoring and surveillance of TB diseasescape as a whole. And right at the end, you touched on the use of the alternative methods or sources of data. Yes. In regards to commercial data coming from fitness apps and things like that.
Absolutely.
So, in your ideal world, which markers would you be wanting to use and measure, and why for TB control? You mentioned something like core body temperature.
We should measure. Yeah, of course, it would be very beautiful indeed to have continuous monitoring of patients, because the treatment is long. It's minimum of 6 months, and There are issues with dual infection, super infection with resistance organisms, and even post-therapy breakdown to disease, which is not uniquely a problem for TB, of course. So indeed, and people are collecting their physiological data with wearables. What we are doing, looking at the host markers, is looking at the immune response to the therapy. People feel better, maybe they get up earlier in the morning, they move around more. So I think, I mean, big data is the story. So we should collect all the data we can. So I'm not going to tell you which one, although I would like, most of the wearables available today do not, for very good reasons, do not measure skin surface temperature, but core temperature. Fever would be a very interesting thing to measure, and not point fever, but fever kinetics, because you know that one of the diagnostics for tuberculosis is night sweats. So and people have low-grade spiking fevers, most probably in tuberculosis, but those high, maybe every 30 minutes or every 20 minutes data for 6 months is really not available. Even when hospitals had lots of nurses, they were not taking your temperature every 20 minutes, they were doing it 4 or 5, 6 times a day. So to have that for a large cohort of patients, for a large population of patients, you could really get those statisticians and the informatics people to mine that data and look for markers. And it may well be that there are indeed kinetics of markers, or who knows what, which perfectly mirror our circulating immunomodulators. So then you would have a non-invasive wearable test. So I would say collect all the data you can.
Excellent, very exciting. Well, thank you very much for joining me, and wishing you all the best of luck with your
It's been a very nice conference. Thanks for the interesting questions. Okay, bye-bye.
Bye.